Hello and welcome to Group A's open source malaria video project. Malaria is an infectious disease caused by protozoans of the Plasmodium genus. Since 2015 there have been 212 million reported cases of malaria, of which 438,000 died. Current treatments for malaria use artemisinin in a combination of other drugs. However, the recent discovery of artemisinin-resistant Plasmodium in Southeast Asia has highlighted the urgent need for new therapies. In 2010, GSK released the details of 13,500 potential anti-malarial medicines into the public domain, and open source malaria was launched the following year. The OSM uses 6 main principles of open source research. The first is that all data is open and all ideas are shared. The second is that anyone can take part at any level. The third is that there will be no patents. The fourth, that suggestions are the best form of criticism. And the fifth, that public discussion is much more valuable than private email. And finally, that an open project is bigger than and not owned by any given lab. This transparency means there's less room for error. In our malaria investigation, we looked at the triazolopyrazines, which consist of 2 main rings with nitrogens on them, as you can see below. There are also 2 spots where we can substitute in various organic groups that make the drug do various different things. Early tests were performed in vitro experiments, that is, they were tested in cells outside the body in petri dishes. However, we need to learn how to test them inside humans, because it's a major problem of these drugs that they're often broken down in the stomach before they get into the bloodstream and start hurting the plasmodium. So we were interested in this particular type of molecule, which had the basic triazolopyrazine core, but had a caged alcohol added on the top. As you can see, there's a cage being drawn now. On the other side, we had a benzene ring with 2 chlorines attached to it. This was the molecule that we were interested in synthesizing. The way we did this was first a condensation reaction, which meant that we started with 2 basic products and then got rid of 2 H's and an O, essentially a water, to form our final product. So you can see we're drawing both of the products now. On the left, we have the double nitrogen part of the core, and on the right, we have the 2 chlorines, which you can see before. There's the water being removed, and we're forming the final product, which you can see being drawn now. The next reaction is known as an oxidative cyclization. The reason it's called that is because we're creating a cycle out of the previous product, which was quite spread out. We're creating the second 5-membered ring. So we're going to copy that in again and redraw it with an extra 5-membered ring cycle, looking almost like the final product I showed you before. The only thing we have to do now is replace this chlorine with the caged alcohol from before. So we're just going to chuck that in on the end, and we're done. Now, to check that we were actually making new substances, we had to use something called a TLC, thin layer chromatography, a layer of aluminium with silica placed over the top. This was dunked inside a beaker filled with a mixture of ETOIC and hexane. We drew a line, put some starting material and some products. As the level of the solvent started to rise, it would drag the starting material and the product with it. Depending on how polar they would be risen to different levels. That way we know that a new product has been formed. In order to achieve disease elimination, drug discovery for the next generation needs to focus on blocking disease transmission and on targeting the liver stage forms of the parasite. An important barrier with respect to the elimination of malaria is the liver stage of the parasite life cycle, because P. vivax can remain dormant in the liver as hypnozoites for many months or years. The only way to deal with this is a new type of drug called tefenoquine. Now, there are some problems with this, however, because these drugs are usually delivered via an injection. Injections can have contamination, for example, and they can also hurt, which decreases the level of uptake in especially marginalized communities. Skin patches are much better. They increase compliance because they require little to no expertise and don't hurt at all. Therefore, we did it. We created the drug we set out to make. But there's so much more to do. There are 13,500 more drugs to test, and we're just getting started.